to examine the antibacterial activity of carbapenem antibiotics on Pseudomonas aeruginosa, a strain of the bacteria susceptible to carbapenem antibiotics is cultured for 24 hours, after which it is spread evenly on an agar plate. Antibiotic-containing discs are placed on the agar plate. The carbapenem antibiotic prevents growth, and the transparent inhibition zone forms, within which colonies appear. Mutants have survived without being affected by the antibiotic, and they have begun dividing and multiplying. Pseudomonas aeruginosa is the major nosocomial pathogen in serious intractable infectious diseases for immunocompromised patients. It demonstrates low susceptibility to many antibiotics, and mutants emerge during division and multiplication. So the treatment of infection by Pseudomonas aeruginosa is quite difficult. Mutants appear at a constant frequency when Pseudomonas aeruginosa divides and multiplies. By exposure to an antibiotic, the susceptible strains are killed. But if a mutant acquires resistance to that antibiotic during growth, it will not be killed and continue growing. This is called the selection pressure of antibiotics. Carbapenem antibiotics enter the bacterial cell through a channel known as D2 porin on the outer membrane of Pseudomonas aeruginosa, then binds to penicillin binding proteins, PBPs, which are cell wall synthesis enzymes, to inhibit its function. As a result, the bacteria cannot synthesize its cell wall and starts bacteriolysis. Resistance mechanisms of Pseudomonas aeruginosa to carbapenems include a lack of D2 porin, an overexpression of efflux pumps which take the antibiotics out from the cell, and a production of carbapenemase which hydrolyzes the beta-lactam ring of the carbapenem to inactivate form, among others. Carbapenem-resistant mutants of Pseudomonas aeruginosa have been found with a lack of D2 porin, an overexpression of efflux pump, and it has been reported that the selection pressure on these resistant strains are different among carbapenem antibiotics. Discs containing Doripenem, Meropenem, or Imipenem were placed on an agar plate to examine the differences in selection pressure on mutants. Approximately 5 hours later, inhibition zones begin to form. Continuing the observation, colonies appear within the inhibition zones. Colonies have barely appeared within the Doripenem inhibition zone. The plate has been spread evenly with Pseudomonas aeruginosa cultured for 24 hours, so the number of mutants within the inhibition zones should be about the same statistically. However, these were the kinds of differences observed in the appearance of colonies. Colonies barely appeared within the Doripenem inhibition zone. Doripenem inhibits the growth of carbapenem resistance. We decided to observe the antimicrobial activity of this carbapenem antibiotic under a microscope. We use agar plates, each containing 1.5 MIC, in other words, 0.375 micrograms per milliliter of Doripenem or meropenem. Pseudomonas aeruginosa cells steadily undergo bacteriolysis as the result of the effect of the carbapenem antibiotics. This is 3 hours later. We expand this field of view. This is the plate containing Doripenem 24 hours later. No cells can be seen multiplying. The appearance of carbapenem-resistant mutants has been suppressed by the Doripenem. This graph shows the results of an investigation for the frequency of emergence of resistant mutants upon exposure to carbapenem antibiotics, using 10 clinical isolates of Pseudomonas aeruginosa. The frequency of resistant mutants was low on the plate containing Doripenem. In other words, this indicates that selection pressure of Doripenem to resistant strains is weak. Likewise, this graph shows the frequency emergence of Doripenem and imipenem-resistant mutants. The results reveal that the frequency is lower in Doripenem. This is a diagram showing the susceptibility correlation between Doripenem and meropenem in Pseudomonas aeruginosa. Doripenem shows strong antimicrobial activity against Pseudomonas aeruginosa. Furthermore, as it is shown in blue, it has been observed that there are some strains which do not show cross-resistance to Doripenem and meropenem. This diagram shows the correlation between Doripenem and imipenem. Doripenem demonstrates stronger activity than imipenem. In this way, Doripenem has excellent antimicrobial properties against Pseudomonas aeruginosa. So Doripenem is a promising option for an effective treatment of infectious diseases caused by them. Next, we will take a look at a morphological change in Pseudomonas aeruginosa treated with carbapenem antibiotics. In the treatment of infectious diseases, samples collected from patients are examined under a microscope to ensure the proper choice of antibiotics and their effectiveness. This is a micrograph of the urine of a patient with acute pyelonephritis. Among inflammatory cells such as neutrophils, you can see somewhat fatter E. coli stained a dark pink and narrower Pseudomonas aeruginosa. The purple cells are Enterococcus species. This is before administration of Doripenem or meropenem. This is 12 hours after administering 0.5 grams each of the drugs. The filamentous bacteria indicated by the dotted line in the screen on the left cannot be observed in the Doripenem sample. Next is 2 days after Doripenem administration. The filamentous bacteria have not appeared even after 2 days, and the number of E. coli and Pseudomonas aeruginosa cells have clearly diminished. This is after a 5-day treatment of each drug. E. coli and Pseudomonas aeruginosa were not observed in the sample taken after Doripenem treatment. Doripenem is the latest carbapenem antibiotic for injection discovered and developed by Shionogi and Company, with the aim of enhanced antibacterial activity against Pseudomonas aeruginosa. Doripenem has a unique antimicrobial profile unlike other carbapenems, such as its anti-Pseudomonas aeruginosa activity, as the result of having a distinctive side chain at position 3 of the carbapenem frame. This is a graph showing the MIC90 of carbapenems. The antibiotics are imipenem, meropenem, and doripenem. Doripenem has a great activity not only against Pseudomonas aeruginosa and Acinetobacter species, among other glucose non-fermentative bacteria, but also species of Citrobacter, Enterobacter, Serratia, and other Enterobacteriaceae, as well as Streptococcus pneumoniae, Viridans streptococci, and other gram-positive bacteria, and anaerobic bacteria such as Bacteroides species. Doripenem, with its excellent antimicrobial properties, was awarded the Pharmaceutical Society of Japan Award for Drug Research and Development 2011. It has been approved and is sold in many countries throughout the world, including the US and countries in Europe.